Hello, my lovely anatomists and physiologists. Michele here, and our topic for this video is the T cell specifically. Now remember that this is a lymphocyte, which means it's involved in the adaptive immune response, or what we can talk about as specific defense. It is, of course, called a T cell because it is going to be trained and mature at the thymus. So the thymus is the gland that we see located in the thoracic cavity. The thymus is more active in our youth than it is as we get older. So we do see that this is a process that's happening during our childhood primarily. The thymus, remember, is located between the sternum and the aorta. We do see a hormone called thymosin, which is going to kind of stimulate T cell maturation. So let's highlight our hormone. Thymosin. Okay, we will see that these cells are called thymocytes when they are immature T cells. And it's at this stage of their development that we see them build what's called T cell tolerance. And what this is the ability to, I'm going to say, ignore self. So this is my phrasing. What this is really talking about is the T cell is able to sort of recognize which cells belong to the body and which cells are foreign. And the reason I chose to say ignore self is because what we'll see during the training process, those thymocytes that bind to your cells are destroyed, so that the ones who are left, the ones that mature, will only recognize and bind foreign antigens as long as everything goes according to plan. So let's take a look over here at this diagram. We've looked at it before in brief. This is looking at the membrane of the T cell. So we have a phospholipid bilayer, and then we have sticking up from that, we can describe it as a 2-chain protein receptor. And so what we'll see is the bottom part of both chains is described as the constant region, and the top part of both chains is described as the variable region. So let's highlight the variable region, and let's remind ourselves that this is the region. This is the region that contains our antigen binding site. So this contains our antigen binding site. And what we'll see is that every type of T cell is going to produce only one type of receptor, so that each T cell binds to just one type of antigen. So way back, we said that T cells are, we said that T cells make 100 trillion different receptors. And this number is so large to me, it's hard to imagine that you have 100 trillion different types of T cells. And so I said, well, maybe we see that a single T cell can have multiple types of receptors. Right? And so that gives us fewer number of T cells, but it maintains our 100 trillion different types of receptors. But that's not true. So I misspoke. I let my imagination run wild, right? So we do see that you have 100 trillion different types of T cell receptors, and that does mean that you're going to have 100 trillion different types of T cells. When we look at the thymocyte training process, again, what we'll see is in the thymus, these cells are going through a process where they have the opportunity to recognize and bind to different types of protein markers. And if their variable region, if their antigen binding site recognizes and binds to one of your cells, that thymocyte will be destroyed because we don't want them out roaming the body, potentially targeting our particular cells. And so what we'll see happening during this training process is, and let's change our color. So let's say ultimately only those, so we'll have it listed as a mature T cell, only those with antigens. Oh, let me rephrase. Only those with receptors that bind to MHC markers with foreign antigens will become the mature T cell. Okay, so let me say it again. During the training process, thymocytes are given opportunity to bind to different cells. And some of those thymocytes will bind to your cells, and those thymocytes are destroyed. Thymocytes that are binding to the MHC markers holding out a foreign antigen. So thymocytes that are binding to cells that are doing antigen presentation are the cells that are allowed to mature. That's the training process. They pass the training. So they're going to mature into a T cell. So your thymocytes are given these opportunities to bind to different types of cells, and only those thymocytes that are recognizing and binding to your MHC markers holding foreign antigens are allowed to mature. And as part of this maturation process, they further differentiate into what we classify as a CD4 T cell, or what's called a CD8 T cell. And so whether you're a CD4 T cell or whether you're a CD8 T cell determines which class of MHC marker you recognize. So whether you are a CD4 T cell or a CD8 T cell determines which class of MHC marker you recognize. So the CD4 T cell is going to recognize and bind with the MHC2 cells. And remember, this is found on our professional antigen presenting cells, which we can abbreviate as APC. The CD4 T cell is going to ultimately differentiate into a helper T cell. And this will be important in what we'll see as B cell activation. So here we're going to have a very tight relationship between the T cell and the B cell. If instead you differentiate into a CD8 T cell, you're going to recognize and bind to the class 1 MHC marker. And remember, this indicates that you have an infected self cell. And in this case, we should consider like the cell is a goner. If it's infected, there's no way for us to just suction out the virus or the bacterium. There's no way to just suction out that particular pathogen. And so we have to destroy our cell for the greater good of the whole body, right? And so this is partly why we feel so bad when we're sick, is because we're actually targeting and killing our personal infected cells. And so what we'll see here is this cell will ultimately differentiate into a cytotoxic T cell. And this leads to death of that infected cell. Okay. The other thing I want us to take a look at here in this particular video is another diagram from our OpenStax textbook. And what we see here is our protein antigen sort of in the middle. And so it's got its whole complicated primary, secondary, tertiary, and quaternary structure. So we see a very complicated, twisty 3D protein structure. And so this we can consider to be basically our antigen. And then notice that we have 3 different T cells. Each of those T cells, we see the variable region having a different color to represent that the variable region is variable between T cell 1, T cell 2, and T cell 3. And so what we're seeing is the antigen determining, the antigen binding site of the T cell is binding to that particular part of the protein called the antigenic determinant. So what this is telling us is that a single antigen can have multiple antigenic determinant sites, meaning that we can be activating multiple T cells from just a single antigen. So this is giving us some redundancy. This is some important redundancy in our adaptive immune response, meaning if there are some changes to this antigen, if the pathogen makes some changes to this antigen such that one of those antigen determinant sites disappears, that's okay, because in this example, there's 2 other cells that will still recognize it. So we still have the ability to mount an adaptive immune response. Okay. Up next, we're going to look at the specific details of that adaptive immune response. So we've kind of talked a little bit about activation. We talked a little bit about this sort of pattern that we're looking for, where we start out with antigen presentation. We went into a lot of details there. We see that an inactive lymphocyte will recognize and bind. So we've just looked at the details of how that T cell has this ability to sort of recognize and bind. We need to get still into this process of co-stimulation. And then what does it mean for that lymphocyte to activate? What type of cells are being produced, and how are those cells effective at getting rid of our pathogen? So stay tuned for some details about this story. And as always, take care of yourselves and each other.